Hello friends, welcome to BMH Learning. In this video, we are going to discuss about minimum inhibitory concentration by Agar diffusion method. Before we talk about the procedure, let me remind you what is MIC, or in other words, we say minimum inhibitory concentration. MIC is the lowest, or you can say the minimum concentration of a chemical, usually an antibiotic, which is able to stop or inhibit the growth of the bacteria. So in this tutorial, we are going to know how we can test the minimum inhibitory concentration experimentally. And to check the MIC, we are using Agar diffusion method. So in this technique, the antibiotic to be tested is first diluted with water, and this makes a series of concentrations. Then, these antibiotic solutions of different concentrations are applied to agar present in the petri dish. This agar contains the bacteria. Now, the antibiotic can be applied to agar in various forms, like in the form of cups, which can be named as cup diffusion. Or the antibiotic can be introduced into the wells made on the agar gel. Then it's called Agar well diffusion method. And also the antibiotic can be indirectly applied on a paper disc, which is then placed on the agar surface. And this time it's called as disc diffusion method. So whatever method we are choosing, one thing is common, and that is the antibiotic diffusion from these sources into the agar medium. And the diffusion of antibiotic leads to inhibition of bacterial growth in the vicinity of the used source, that is the antibiotic source. The source represents here the spot at which the antibiotic is applied in the agar, and the area up to which the antibiotic shows its effect is referred as zone of inhibition. Antibiotic agar preparation. First of all, let's start with the preparation of stock solution. So, we need antibiotic. The choice of antibiotic depends on the type of microorganisms you want to test against. Usually, it's available as 600 mg per bottle. The 600 mg antibiotic is diluted with 6 ml to make stock solution having concentration of 100 mg per ml. From this stock solution, we take 1 ml and transfer it into the another tube containing 9 ml of distilled water. This would give us the concentration 10 mg per ml. From this 10 mg dilution, we are gonna make 1 ml aliquots. Of course, this would have the same concentration of 10 mg per ml. Don't forget to mark the aliquots with the antibiotic concentration, date of preparation, and expiry date. Store these aliquots at -60 degrees centigrade. Next is the preparation of antibiotic dilutions. Now we are gonna make various dilutions of the antibiotic. So here comes the main point. You need to select the range of antibiotic dilutions needed, and this is based on the MIC range for quality control organisms. So this is going to be different for each antibiotic. Let's use one aliquot having concentration of 10 mg per ml. We transfer 1 ml into the tube having 9 ml of distilled water. The concentration would be now 1 mg per ml. Now, from this 1 mg per ml concentration antibiotic solution, we are gonna make required dilutions. As I already mentioned, the dilutions depend on the MIC ranges for a specific organism. So, you need to make it according to that. But for now, I will tell you, generally these are in the concentrations of few micrograms per ml. Let's suppose, for example, from 1 mg per ml concentration, we take 100 microliter and transfer it into 19.2 ml of distilled water. This would give us 20 ml and the concentration is 4 microgram per ml. From here on, we are gonna double dilute by serially transferring 10 ml from each tube. So for that, we transfer 10 ml to make 2 microgram per ml. And then to next tube to make 1 microgram per ml. And similarly to the next one, which would give 0.5 microgram per ml. And this thing goes on. Next step is the preparation of agar. So, according to the need of the experiment, required amount of agar solution is prepared. The agar solution is then poured into the petri dish. The important thing here is the agar surface is inoculated by spreading a particular volume of the microbial inoculum over the entire agar surface. So the agar solution has the bacteria against which we are testing the antibiotic. Now for the better results, we must prepare separate petri plates for each antibiotic dilution. The petri plates are then left to solidify. Next step is the application of antibiotic on the agar surface. As I already mentioned, there are various ways to do that. So we can either take a paper disc and dip into a particular antibiotic solution and then place it on agar surface in the petri dish. Or otherwise, we can make a well in the agar with a well puncture, and into this well, we add antibiotic solution of specific concentration. The process is repeated for each different concentration. We also keep a control paper disc. It's usually placed for comparison. It's only soaked in solvent. After this, the petri plates are incubated at 37 degrees centigrade. Results and interpretation. First, watch for the control disc. It does not show inhibition of growth. If there is a formation of inhibition zone around the disc or the well, this shows the antibiotic is working against the bacteria. The diameter of these inhibition zones increase with antibiotic concentration. Then we check for the least concentration of antibiotic, which still prevents the growth of the microorganisms. This would be the MIC of the antibiotic used. This was all. Thanks for watching.